I'll probably take.
You did the birthday.
I did ice bath.
Oh, that's no problem.
Sherman, we were all so sore. We were like all in this.
Yeah, just let me know when you want us to start. I think I might just get a quick drink of water.
I don't remember. We get down to like 10 degrees now. Oh, okay, because like here might be a little colder. 10 degrees out of ice.
Thanks, Jake, for sending out that email. Appreciate it.
Well, I mean, I told it, take this, but I don't think one, but ABS next year, you know where it is? Like, should be like, I feel like I've heard, but I don't know. It's in West Palm Beach, Florida. I did know that, I did. And my mom's just beyond excited about it because she's like,
Really, that's awesome.
yeah, she's like, I can make everyone milkshakes. Right, yeah. So,
It's supposed to be in 2020.
yeah, I knew they were doing like the COVID, like that.
Yeah.
And burgers.
Where's the microphone? Yeah, I've got this.
Okay, awesome. Thanks, guys. So I think we've got like a good group of people, number of people attending online, and we've got a great presence in person too for everybody online. Yeah, you kind of missed out a little bit. There's some special beverages in the corner here that you missed out on. But welcome back, guys, to the CNM Nutrition Seminar Series. Really happy this month to have Dr. Mike Steele and his lab presenting on microbial mayhem. So maybe I'll just pass it right over to you, Mike, to introduce and from there.
Thanks, Jen, for the introduction. It's a pleasure to be here today and share with you some of the research that we've been doing related to the microbiome in the calf. And now we're moving into the cow, but this has been an evolution of research. And I wanted to go through some of the ideas, and can't go through it all today, but I also wanted to showcase some of my great students. So, of course, Latero, who will be moving on after this month to go to the University of Wisconsin. The bulk, and this will be his last opportunity to present to the department, so this is kind of special for us. That's why we only have Argentinian wines here, okay? Only that for today. And also Koki Nishishira, he's my postdoc, and he'll be talking about some models he's working on, as well as Francia will also talk about a model that she's working on. So first things first, what I want to go through today is why microbes are important in this interaction between the microbe and the host, okay? So that will be a theme for today, is not just looking at microbial diversity or the community structure, but actually the host, because we think that's most important, especially for what we study. And then we want to discuss some of the first investigations looking at microbial interactions, and this is all with calves. And then we want to talk about some of the stuff we're doing now and also in the future related to transition cows, weaning, and enteroids. So first things first, I'd like to say, first of all, I'm a ruminant nutritionist physiologist. I'm not a microbiologist, but I do have a deep-rooted love of microbiology. And it's been quite some time. It's actually when I started the micrograph in the last slide, when I started characterizing protozoa and different bacteria on the rumen wall, I really fell in love with it. And then I realized there's other people like me out there. And it's the godfather of microbiology, Anton van Leeuwenhoek, from the Netherlands, who started microbiology. And actually, he was a surveyor by trade. He made 500 of his own microscopes. This was a hobby for himself. And he's the first person who saw a microbe in water. He also is the first person who characterized spermatozoa. So I'm a huge fan. I'm also a huge nerd. I know I'm pretty cool. You probably don't know that, but I love this stuff. And actually, I was in Delft in the Netherlands. I knew that this was where he was born and where he died. So I thought there'd be this massive statue, but there was only this one plaque. But it really was quite meaningful for me to be there at this time. Someone that I really looked up to. And this is where you would see in Delft in the Netherlands, saying that this is where he actually was born, also died. And his death was in 1723. So a lot has changed during this time, and we've really become connected to microbes. And actually, a lot of points in human history are reliant upon our microbial interactions. But of course, we enjoy it, and that's why we're enjoying it today. And I just want everyone to appreciate microbial fermentation. Microbes in the cow, we've had this relationship for a very long time. I think that the most prominent example of this is smallpox vaccinations. And vaka in Greek translation is cow. So when you're getting vaccinated over the last 3 years, you're actually getting cowinated, okay? I think that we need to really understand that and really appreciate the cow and also the microbial interaction between humans and cows too, because it plays a big role in our history. Of course, microbial fermentation, these are more micrographs where I fell in love with microbes during my PhD. So this is actually some cockeye colonizing or chewing away and actually fermenting a piece of hay that's attached to the rumen wall. So I just want to say that there's lots of economies that are totally based on the rumen, okay? So New Zealand is a classic example where most of their GDP is totally based on the rumen, and we have a big ruminant industry here that's approaching around 20 billion. So it is really important for our economy. Now, things have changed a lot. So when I started my PhD, there was no next generation sequencing. I recall being up in Brian McBride's office in about 2009. We had a conference call. We didn't have Teams at that point. We didn't have video conferencing when I was a PhD student. We called the person, he was at Vermont, and he said, we were characterizing using DGGE, which is basically just fingerprints of the DNA to establish their microbial community or to learn more about that. But he said, we cannot do this anymore. Every single thing from here on in, so this was 2008, 2009, we have to sequence everything or else we can't publish. So this really happened quickly. We didn't really know much about these technologies. They were at a very high level. We were just looking at phylum, genera, maybe a little bit of family at that time, but we were characterizing quite a bit. And I think this is where it really took off with respect to the human research. So we probably all have heard of Jeffrey Gordon's group. One of his PhDs, who's currently at Cornell University, was the first person to characterize in humans from birth all the way through their life. So they were sampling sometimes the fecal microbiota almost weekly in some of these patients, and then determining what is actually imprinting their microbiome, which is kind of neat. Now, there's lots of things that can change, that can do this. There's still this big debate about colonization prenatally. Not a lot of evidence from what we can see, but also the delivery method, C-sections, milk feeding, breastfeeding, infant formulas. What does that do? Also the environment and the complexity of this environment. And this was at the same time when all these papers were being published. I was doing a bunch of cow work, but I thought, wow, this is a fascinating area. And it happened to be the same time of when I was growing a family with my wife. And it was interesting because my son received a lot of antimicrobials in early life. Granted, I don't have a lot of degrees of freedom here, but my daughter didn't really receive any. And I can tell from their gut health that perhaps this might be a contributing factor. But this is where it got me thinking, wow, we do things totally different in the infant and human world compared to the bovine. And there's several examples. I don't think we really understand in the bovine how the delivery method of C-section versus a natural birth really affects the calf's microbiota and potentially for life. Also the milk feeding, whole milk versus milk replacer, different milk replacer compositions. I don't think we really understand this. And same with the interaction between the dam. And also the environment. Our calves have been raised in isolation for the last 40, 50 years. And now we're breaking out of that and doing more group housing. So all these things are happening. We know that in other models that they're changing the microbiota and even for life. So I think there's a tremendous potential here to learn more. And the group that's doing most of the work, and this is the first group in the early 2010s, was a PhD student at the time, Nilu Malmathuj, okay? So she's currently an a research scientist at Agriculture Canada in Lethbridge, and she was doing her PhD with Professor Leilu Guan. Now, this graph just shows the huge growth in the literature during this period. So early 2000s, hardly anything there related to the rumen, but all of a sudden it takes off, especially with colonization of the young animal. So this is a picture of Dr. Malmathuj, who we work with all the time in this area. So all the students work with Nilu as well as Professor Guan, who's, I just wanted to state, these two have contributed a tremendous amount. There's no way I would do this research without amazing collaborators like them. But they've done the most work, and they've actually characterized in rumen fluid and fecal, just looking at it temporally, what can change during this time. But now it was wonderful partnering with them and to look at how some nutritional interventions can influence a lot of these, a lot of the microbial communities in a calf. And why this is important, because calves are still dying at a really high rate, okay? So mortality is still between 5 and 10% during the pre-weaning period. And a lot of this is coming from digestive diseases and disorders. In fact, diarrhea is still the biggest issue in the calf world. And it's probably linked to respiratory disease later on. There's a lot of hypotheses related to that. But it's something to take note of and lots of opportunity to improve there. And also, I think we overuse antimicrobials. Sometimes we'll have a protozoal infection and we'll go straight to an antimicrobial. It doesn't really make a lot of sense. I think we're becoming a lot more prudent in the dairy industry as well as the beef industry, but I still think there's a lot of work to be done, and we'll talk about this in this presentation. So why we're interested in this is calves treated with antimicrobials during the pre-weaning period have lower conception rates, increased time to first calving, increased risk of culling, as well as reduced milk in first lactation. So this is something to pay attention to. I think that we are overusing it. Obviously, from a consumer acceptance, it's a big issue. But even for the calf welfare, I'm not sure if we're really promoting welfare in most circumstances. So I'd like to pass this time and discuss one of our first studies looking at the use of antimicrobials. But I'll have Latero, who just graduated last month with his PhD, go through this experiment. So the floor is yours.
Yeah, thank you, Mike. So as Mike was saying, we were really interesting to see the role of antimicrobials have on gut health of these animals and on metabolic function. So the first question that we wanted to answer is, there's a lot of prevalence of the use of antimicrobials prophylactically on these calves to use as a preventative for some of the gut health issues these animals have during the pre-weaning period. So, but there's not a lot of evidence supporting the efficacy of this practice. So the first thing we wanted to see is actually what is the response that these calves have, specifically with neomycin. That's the most prevalent antimicrobial use prophylactically on calves. And then from there, try to build recommendations from the industry. So we had an experiment with 160 bull calves in a Ville facility that is close from here. And then these calves, we distributed between a control group that did not receive a medicated milk replacer, and then two groups that received neomycin at 20 milligrams per kilogram of body weight, either for 14 days or for 28 days. And then a subset of those animals were dissected on day 28 of life. And then we also did gut permeability tests on these animals. So in addition to that, something that is very recurrent that we see in several animal models and in humans is reduction in microbial diversity, specifically with the use of antimicrobials, can result in dyslipidemia and increased fat deposition. And in humans, this has been seen as one of the risk factors for development of metabolic disorders. And in farms, antimicrobials, not as much in calves, but in pigs and in poultry, antimicrobials are routinely used to improve efficiency of gain and fat deposition. So one of the things that lately has been an interest for us based on other studies is to see if changes in bile acid metabolism has something to do with these changes on the rate of lipid and fat deposition, based on early studies from Dr. Iparraguirre, that he was a committee member in my PhD, and why we were interested in bile acids. So bile acids, most of you know that the main role that they have is emulsifying lipids after ingestion of a meal. And they also have antimicrobial properties, and they prevent overgrowth of microorganisms in the gut after ingestion of a meal. But then there are some particular bacteria that has the ability to transform these bile acids into what is called secondary bile acids. So these bile acids are synthesized on the liver and they're secreted after ingestion of a meal. They help digest those lipids and emulsify those lipids to improve digestion and absorption. And then there are some microbes that has the ability to transform them so they are not as toxic to them. And then because these molecules are very pleiotropic and they can act across the body at very low concentrations, then this system is tightly regulated in the enteropathic circulation by specific receptors. And then one of the most interesting functions that has been shown recently is that some of them can escape into systemic circulation and they act as nutrient sensing molecules. So they convey information from the intestinal environment and they alert peripheral tissues that incoming nutrients are coming. So some people are starting to wonder what happens when there are changes on the microbial composition on the gut, if that affects the changes on bile acid function, and if that can be a mechanism that can answer the changes on metabolism on some of those cases where we have dysbiosis. So we wanted to look at first how this neomycin was affected growth and health of those animals, if there was actually any benefit in terms of performance of those animals and on gut health. We actually didn't see any effect improving average daily gain of these animals through the pre and post weaning period. We also saw a small reduction just on fecal scores during the time when these animals developed the most incidence of diarrhea. But then days to first diarrhea, and also we also look at resolution of diarrhea wasn't changed by the introduction of neomycin in the diet. So based on this data, there's not a lot of support to actually recommend this practice. But then some inadvertent effect of this practice is that it reduces microbial diversity on that gut during the pre-weaning period. And this like shown in here by this index is Cal1 is an index of species richness, and phylogenetic diversity is the overall diversity of that microbiome. And it acted on a location-specific way, so that the ileum was the location that was like mostly affected on these animals by the use of neomycin. Then, because neomycin, the microbes has a very important function in maintaining the integrity of the gut wall, we wanted to see if the use of antimicrobials throughout this period was affecting intestinal permeability, and in fact, the use of this neomycin increased intestinal permeability on these animals during the second week of life, which coincides with the time that these animals have the highest incidence of diarrhea. And then from there, we wanted specifically to look at the relative abundance of bacterial genuses that we know have the enzymes to metabolize these bile acids and transform them into secondary bile acids that I was talking before. And we saw a pretty significant reduction on lactobacillus, bifidobacterium, both in the digesta and in the mucosa, both in ileum and in colon. So then from there, we pose the question, okay, so if these bacteria are reduced due to the neomycin, what's happening with the bile acids? So we look at bile acid composition both in digesta and in tissue on the intestine in several locations. Here, just to make it simple, I'm just showing the tissue where we found the response. And what we saw is that both short-term and long-term treatments with neomycin increase the reuptake of bile acids, both primary and secondary bile acids in the ileum. And if you remember what we saw before, also the ileum was the area most affected on microbial diversity and relative abundance of those species. And then we also look at plasma, and then what we saw in plasma was the opposite, is that these secondary bile acids, the ones that results from transformation of microbes, they were reduced in plasma. From there, we want to see, okay, if there was a reduction in plasma, is this translating on changes on adipose tissue function? That is one of the main organs that these bile acids target. And we ran a chip of 48 different genes that are related to lipid metabolism, glucose metabolism, and immune function. And here I'm just showing the ones that were differentially expressed. So we are cautious with this data just because only 5 of all the 48 different genes were differentially expressed. And this GPAT and lipid-1 that are encoded for triglycerol biosynthesis, FBP1 that is a rate-limiting enzyme for glucose synthesis, and then these genes that are related to immune activation macrophages were changed by the short-term group, but not the long-term group in here. So in conclusion from this data, we believe that this prophylactic use of neomycin might be implicated in this regulation of this bile acid metabolism, altering its profile and tissue distribution. And then these changes in transcript abundance might be related to changes in metabolic function and adipose tissue, but this is still on the works, and we need to do a lot more work to actually be able to determine or say if changes caused by antimicrobials are affecting metabolic function as a whole in the animal. And we would like to see, and this is something I will try to see in my new faculty position, is if these changes in early life in adipose tissue function also are sustained over time, and if these mechanisms can be at least partially responsible on the changes that we see at the cow level that we saw on the epidemiological data that Mike was showing of cows receiving antimicrobials in early life affecting performance of those animals when they reach their productive life. So then from here, I'll pass it to Mike.
Great.
Yeah.
So yeah, this will be a lot of Latero's work moving forward. Will be in this area where we really need to follow these animals for a lot longer. It's one thing to do these experiments during the pre and maybe the first post-weaning period, but in order to understand the true implications, we have to follow it. So kudos to you for wanting to do this. Now, we need to also look at alternatives for antimicrobials. There's lots of countries where antimicrobials are being phased out, and I think eventually we'll be in the same position. But I want to keep in mind that actually the probiotic world in the animal industry is growing, but it's still quite small compared to the rest of the industries for probiotics and other applications, but it's still quite important. Now, Latero also wrote a review, and here's some of the results from that review, just looking at bacterial-based probiotics as well as yeast-based probiotics in calves. And I think when you really look at the literature, all we're really looking at is the phenotype of average daily gain and health, and through health scoring. And I think there's this strong need to understand that a little bit more, because the same probiotic in different environments will have a different response. And in order to tease that out, you have to understand the mechanisms more, which led to a series, and this was the first time, so I was a new professor at the University of Alberta. I had the opportunity to work with Lallemand, and they gave me some funds to do a really simple experiment. And this was my first exposure to the probiotic research when I was an independent researcher. I was very thankful that they provided this one probiotic by the name of Saccharomyces cerevisiae boulardii, which you can actually try right here. These are very safe and will actually have been proven to improve your health as well. But what was really intriguing about this particular yeast is that it can affect barrier function of the gut, which is something that we really wanted to focus on, and also the immune response. So it's one thing, like, of course, looking at the microbiota and characterizing that, but that wasn't really our focus. So we started simple. This was my first study, and I failed. I really didn't do a good job on this one because we had incredibly healthy animals. So we found zero response. This was done in an actual farm here in Ontario. They were extremely clean. They would actually take all their feeding equipment and put it in -20 because that's how scared of crypto they were. So just really amazing hygiene on this farm. And what happens when you do this is you don't really see a response. Okay, so we very quickly, in the first year of my professorship there at the University of Alberta, we said, this is it. Like, if we're going to actually study probiotics, we can't just dump it into healthy animals. We need a challenge model. So we went back and forth on challenge models. We were thinking of E. coli, crypto, rotor and coronavirus as well. The problem with all those is you're just looking at one pathogen. In your replication, it's very rare to see a disease challenge model with over 15 animals per treatment. So we knew that was going to be criticized, and we wanted to use a more economical natural challenge model, being in the veal industry. So we did an experiment at Grover Animal Nutrition when they had their veal calves there. And through feeding Boulardii through the milk, what was happening is your fecal score was being reduced during the time of diarrhea, as well as the diarrhea cases also reduced. So this was interesting. At least we had our approach right. We did sample the bacteria, and we also sequenced the DNA from the fecal samples. And again, we didn't find any differences. With even large replication, very little differences. That might reflect what's actually happening. It might be a limitation of the actual 16S sequencing that we were doing at the time. But we really didn't find anything, which led to recruiting the most energetic postdoc I've ever had in my life. She would actually run marathons a lot. The first 24 hours of her being in Canada and Alberta, she actually climbed the highest mountain and took a picture of herself doing a handstand. This was a very, very high energy postdoc by the name of Clotilde Villot, who did a MyTax with Lallemande. And now she's a research scientist and works for Lallemande in France. So this was a lot of her ideation. So I just want to make sure she receives a lot of credit for this. But we had these results and we said, well, we got to figure out what's happening. And we think it's with the host. We know that there's hypotheses based on feeding Boulardii and affecting gut function, primarily increasing IgA production in the gut. So we wanted to figure that out more. So as soon as calves were born, they weren't born in the pen. They were born, we would minimize dam contact. There would be no exposure to the dam outside of the birthing process. And then we immediately put them on this probiotic and we followed them in great detail for the first week of life. And what we were finding, and I'm going to give this to Latero, who will be going through this in greater detail, is that actually, and it was so funny watching Chloe do this because the first time she saw this result, she's such a critical thinker. She said, can't be true. No, we're going to repeat it. Going to do it again. Yeah, it's true. Okay, we need to look at the B cells within the gut just to confirm this IgA result where Boulardii was increasing IgA within the lumen of the gut. Turns out, yes, the B cells were at a higher population in certain segments of the gut. So yeah, it looks like based on the literature and based on our findings, this is a legitimate hypothesis that we really need to keep on testing. So actually, Latero took part of this for his PhD, and I'm just going to have him go through how he excelled this study even further.
Thanks, Mike. Let me put this color on. There we go. So yeah, as Mike said, I was very fortunate to be able to work with Chloe and the Lallemande team on this experiment. And basically what she did, she had 20 calves, one group receiving the Saccharomyces cerevisiae boulardii every day for 7 days. And then the other group just received a carrier that was a control with no yeast on it. And those animals were dissected on day 7 of life. And then they measure concentrations of secretory IgA, the different compartments on the gut mucosa. And then what they saw is that the probiotic treatment was able to increase secretory IgA production on those animals in the ileum and in the colon. So from there, we wanted to see what was the Saccharomyces treatment doing on the microbiota of those calves and see if by explaining that part, we could explain the role that it had in increasing humoral responses. So that's what we did. We sequencing samples of the different compartments on the gut, both in tissue and in the digesta. And what we saw, and here I'm just showing digesta of these animals, that the animals in the same sections that we saw an increase on secretory IgA, there was an increase on species richness, at least a tendency on the colon for that, and then an increase in phylogenetic diversity on the ileum of those animals. And then to look at this microbial population a little bit further and try to understand how it was improving maturation of this community, we use network analysis. And like the network analysis, what it allows us to do is to have a visual representation of that ecological community and how they interact with each other. So what we saw is that the group of calves that received that probiotic, they had increased diversity, yes, but also they were way more interconnected with each other, which are attributes of a more mature ecological community. And we were able to see that by using this network analysis. And then from there, we say, okay, from the most differentially abundant group of bacteria that we saw between the Saccharomyces and the control group, let's do a correlation to try to see how that changes on diversity, on relative abundance is related to the secretory IgA and then BFA concentrations and diversity. So what we saw is that, yes, these individual bacteria increasing this relative abundance on the Saccharomyces group were positively correlated with that increase in secretory IgA in the ileum, and also were directly correlated with those increases on microbial diversity, okay? So this is just a correlation, and we know this is not Causation is really hard in ruminants to be able to actually be able to discern and depict or dissect these differences. But this brings us a step closer trying to understand what this is actually doing on the gut of the calf. So in conclusions from this, we saw that the supplementation of these yeast increased microbial diversity, increased the community interconnectivity and stability that are attributes of more mature microbial population. And then we think that because of this is what provided the necessary signals to stimulate the increase in secretory IgA production and secretion. And then future studies, we want to focus on also the long-term effects that this has. If we supplement it in early life, how those animals respond later on in life. Then I think, yeah, there you go.
Yeah. Now I'd like to talk about some of the models that we're currently using and we hope to use in the future. But this is an experiment that we've wanted to do probably for 6 years, and we're really thankful that we have this opportunity to do a transition cow study with the same yeast here, the same Boulardi yeast. So this is something that we're doing right now at the Elora Dairy Research Center. So very simple experimental design. It's either supplementation with Boulardi or no supplementation. It's throughout the entire dry cow period, the late gestation period, as well as through early lactation all the way to 70 days in milk. So the big take home I want to show is that we think it's really critical to characterize the host. In fact, characterizing the microbial community was kind of secondary in this study. Of course, we have to do it. We will do that for sure, but it's not the primary outcomes at all. As you can see, we have lots of sampling looking at, for the first time, 80 cows through transition, looking at gut permeability. Also doing a lot of flow cytometry, not just on blood, but also the colon biopsies, which we'll discuss later. So we want to make sure that and take it to the next level where we're characterizing the gut barrier function, also the immune system in greater detail. And we want to see how this relates to the microbial communities as well as the interventions of Boulardi that we're providing. Another aspect of this experiment, and this comes from experiments that we were fortunate enough to collaborate with Lallemand on in France, where they're feeding yeast probiotics to use, and it appears to affect the composition of colostrum, which is really an intriguing idea. So we're looking to do this and do a little bit more experiments, not only on the colostrum, but there's some evidence that the yeast, even fed prenatally, can affect the offspring. So this is why we're characterizing this in great detail. This is a tremendous amount of work to do this kind of stuff. So kudos to all the students that have taken this on. This is one of the most intensive studies I've been told at Elora. So we thank Elora for letting us do this. But yeah, definitely, we only have 10 calves right now. We have 80 more to go. So we'll probably be at Elora for the next 6 months. And special thanks to everyone. I noticed Tony's here. Thank you for all your help prepping and everyone else who was involved in this study. Now I'd like to go back. So that's one model. We've always wanted to do it. I think actually the transition cow model is very similar to the weaning model. So there's many times when our lab has studied weaning, but I think a lot of the results can be transferred to the transition cow. So we've been studying weaning for 9 years since I became a prof. It's been something that we've always studied and characterized in different models. But we wanted to see how a probiotic, and this was the first probiotic research that we looked at, purely using weaning stress as a model. And we wanted to look at lactic acid bacteria, and this will lead to what Ferencia is about to tell us. But there's a lot of data on lactic acid bacteria in calves. We're putting it in the milk predominantly, and it has been affecting just the phenotypes of health as well as average daily gain. There's not a lot of evidence like what exactly is it doing to, or what are the biological mechanisms behind that phenotype. And this is where Ferencia's MSc and also some of the work that Latero did during his PhD. And also I want to mention, I noticed that Brooke was online. She was also a really big part of this experiment as well, looking at the gut-brain axis, which we're not going to talk about today. But perhaps I'll just give it to you, Ferencia, to describe this model in greater detail.
Okay. Thank you, Mike. So we ran this study with Lautaro and Brooke last year at Ponsonby General Animal Facility, where we were looking to evaluate if the supplementation of Lactobacillus helveticus in milk replacer during the pre-weaning period of hosting bull cows can ameliorate the negative effects of the weaning process on the barrier function and the markers, the inflammatory markers response. So we enrolled 44 cows, 22 in each treatment, the control treatment, and the tendalized Lactobacillus helveticus, which is a heat-treated bacteria. And we apply an abrupt weaning model where we switch from a predominantly liquid diet to a solid diet in just 5 days. And we also perform gut permeability tests before and after the weaning period. And we also took blood samples to measure serum amyloid to evaluate the inflammatory response. Specifically, these blood samples were taken on day 35, which is at the beginning of the weaning period, and during the weaning period, day 42, 49, and 56 as the post-weaning period. Yeah. So as some preliminary results, we have got permeability results. We didn't find any treatment differences in here, but we did find a difference by day, showing that the pre-weaning concentrations of the gut markers were lower than the post-weaning period, as you can see here in this graph, where we show the chromium AUC, lactulose AUC, and the lactulose mannitol ratio, showing that we kind of perform with this abrupt weaning model and a damage in the structure of the gut. So for the serum amyloid A, we wanted to go further, evaluating the impact of the weaning process, and we found a different, a treatment by day difference, where on day 35, 42, and 49, where the control treatment had higher values for serum amyloid A, which is showing us that the TLH supplementation may help regulating the inflammatory response after these processes. And this is where I'm gonna work further on my program. So now it's your turn, I guess. Oh, it's your turn.
Thanks. So yeah, like one of the things that we're interested to understand is like not only the changes on the microbiota, but what is actually the impact that changes on microbial composition has on gut health and immune function, specifically on the mucosa of the gut. So we developed this technique that allows us to isolate lymphocytes from colon biopsy samples and allows us to do longitudinal studies to understand how changes on the microbiota is affecting mucosal function. One of the limitations of this technique that we can only reach the colon, so we cannot really extrapolate what's happening on the colon to the rest of the gut, but at least it gives us a pretty good idea, and then we can follow these animals throughout time without the necessity of putting a cannula or dissecting these animals to be able to reach these samples. So then we isolate these lymphocytes, and then we can stain with fluorescent antibodies, and we run in flow, and we look at cell function at specific populations. And we were really interested during my PhD on this group of cells that are gamma delta T cells, because they are at high numbers in early life, and they confer some protective and regulatory function until other branches of the adaptive immune system start to kick in. So I'll just show you the results on, I'll just skip this because of time, on gamma delta T cell proportions in the colon from day 2 of life until weaning. And these are called Tisney plots, and it allows you to cluster cells based on the different criteria that we use. So are roughly 1 million cells that each of the cows that were enrolled in the study are contributed equally to this plot. And then we can separate or do clusters based on certain characteristics. So as in general, lymphocyte populations go from 3% or 3.5% at birth all the way to close to 16 when those animals are weaned. So like it gradually increases over time. And then these gamma delta T cells populations, they don't change in proportions during that first months of life, but after weaning, there's a dramatic change, an increase on gamma delta T cells in this location. We think that partially because of weaning. Okay, so flows show there was an increase on intestinal permeability, and maybe these cells are kicking in here to respond to that challenge. And we also look, I don't want to go into much detail on that, but we look at the different subset of populations of these gamma delta T cells that are mainly divided into two that has effector function and regulatory function, and they both went up during this time. And then wanted to see, okay, how weaning is affecting microbial diversity throughout that first month and a half of life of that animal. And then from day 2 to 28, there's an increase on microbial diversity, species richness, and phylogenetic diversity. Well, not in phylogenetic diversity, actually. And then from day 28 to 42, that is when that weaning is happening, there is a dramatic reduction in microbial diversity on these animals. So from that, we wanted to try to correlate how the changes on microbial diversity that we see here from day 28 to 42 correlates with those changes on immune function that I just showed you before. So we did a Spearman-Grant correlation analysis between those immune cells that I show you and microbial diversity. And yes, these gamma delta T cells are negatively correlated with microbial diversity. So as microbial diversity is going down, these cells are increasing either by migrating from circulation into the gut or by expanding as a response of that weaning challenge. And also these subsets are negatively correlated with phylogenetic diversity. So in conclusion, and again, this is just correlation data. We kind of like really tell for sure there is actually a response of the microbiota. It could be just ontological adaptations that are happening, but this is coordinating this gamma delta T cell expansion. And the abrupt weaning is reducing microbial diversity and promoting this increase in gamma deltas. So Mike, pass it back to you.
Awesome. So the last part, we just want to show you, those are a lot of the in vivo models that we're working on. So lots of weaning and lots of transition cow work, hopefully in the future, is in addition to the pre-weaning work. But I wanted to, a lot of the things that we're doing, as you can tell, are very intensive. And we're not allowed to do a lot of the stuff like fistulations. We were just declined. So I think I'm seeing the writing on the wall that we're going to have to move to in vitro to answer a lot of the questions. So when looking at in vitro models, a lot of them are primary cell cultures or just one cell type. It's very unique to see a co-culture in some of our animal science fields, but they do exist. But we're more interested in entroid models. We find this a lot more exciting and closer to the actual animal itself. So I've invited my postdoc, who's currently in Japan, by the name of Koki. He'll go through this work. It's actually almost 3 a.m. in Japan, so just be aware. But we want to thank Koki for coming online, and the floor is yours, Koki.
Okay, thank you for introducing. So entroids can be a variable tool for analyzing the effect of probiotic on gut function. However, some people maybe wonder what entroids are. So I would like to introduce this article first. So last year, a medical team in Japan successfully transplanted mini gut into a patient with colitis. These mini guts are entroids. Next, thank you. Entroids can be used for not only patient treatment, but also fundamental research. They are useful for analyzing the interactions between hosts and microbes, and have been co-cultured with pathogenic bacteria and virus in several studies. And I'd like to explain why entroids are useful for us. Entroids maintain a three-dimensional structure and cell population similar to the original organ if we compare to traditional two-dimensional cell culture models. And they can also be proliferated and cryopreserved like traditional two-dimensional cell culture models. Therefore, we can replicate them and reduce the number of animals used in vivo models. And development of entroids is not difficult. Enteroids can be cultured by isolating crypts from the intestinal tissue and embedding crypts in a specific gel. Our group has successfully developed enteroids from both fresh and frozen tissue, which allows us to study enteroids from samples taken at farms which are geographically far from a laboratory. After enteroids are cultured, they proliferate every day in the specific gel. There is no significant difference in morphology between enteroids from fresh and frozen tissues. And I also want to share the other images of enteroids. Some enteroids show a budding structure that resemble crypts in the original organ-like left image. Also, immunofluorescence staining has shown that brush border marked in red in center and right images distributed inside enteroids, like in the original intestine. The other group have shown that enteroids have similar transcriptome to the host gut. This means that enteroids are useful tools for us as in vivo models. And our future plans are culturing enteroids with probiotic and studying their effect on intestinal function, such as barrier integrity, nutrition uptake, and immunity. We also plan to culture enteroids and probiotics with lipopolysaccharide or at higher temperatures to mimic heat stress. And we want to study the effect of probiotics under these conditions. Thank you.
Yeah, thanks for coming in at close to 3 a.m., Koki. Really appreciate that. So I just wanted to firstly thank everyone, but just to let you know, the main take-home messages here is that there's lots of interaction happening. I think where we've kind of fallen behind is our phenotypes are just growth as well as health scoring. And I think we need to look more deeply into the host and not just the microbiota. The importance of a challenge model and having that challenge in order to study essentially many concepts, but especially probiotics, I think is really important. And also characterizing the host in great detail. And that's what we're doing as a lab. And we're really excited about a lot of these new models. Now, I know we've been doing weaning, but I still think it's a really effective model. You look at piglet research, a lot of the probiotic or prebiotic work is on weaning. So it's a very effective model. Also transition cows, but enteroids, I think, will let us answer a lot of our fundamental questions efficiently. And really thank Koki for developing a lot of those techniques for us to use in the future. So I'd like to thank everyone that's contributed to this, all the graduate students, but especially Lallement, who's been funding this branch of research since the beginning. Now Lanco and also the Dairy Farmers of Ontario and Alberta Milk and NSERC and also the provincial government. But a special thanks to the presenters, and I want to say goodbye to Latero. This will be the last time that he presents as a student too, or like I guess you're not a student now, a member of our department before he goes to the University of Wisconsin as a professor. So a lot of this work today, obviously, I think this is the last time I could really call upon Latero to present, and I totally took advantage of that too. But as you can see, he contributed a lot to this research, so I wanted to make sure that he got the proper acknowledgement of that. But also, thank you so much, Florencia, and thank you, Koki, for sharing a lot of the data that you've been accumulating over the last year. So very happy to take questions. Thank you. Yeah, any questions?
Yeah, any questions online or in person for Mike?
Question is for Koki. Can you measure permeability directly with enteroids? So Koki, this is from Professor Giselle Lapointe in food science.
Directly, maybe no, but we can culture enteroids with fluorescent, like FITC, and we can capture the image how fluorescent embed inside of enteroids. And we can also develop 2D enteroids like and we can measure how to say in English, TER. It's the value for gut permeability in vitro model. Thank you.
Yeah, so there's ways of doing it with the whole Android, Professor Lapointe, but a lot of people are just cutting them up and creating a 2D, doing it 2D after they've grown the Androids. We'd like to try both, actually. And this is like, I just want to say this is something that I thought, and we were told like we weren't able to do. Like we were told by the people who started, who have been working on this, that like be ready for 2 years of failing in the lab. And kudos to Koki, he really figured this out quickly. So we're ready to do some studies as soon as he comes back to Canada. Just in time for the Raptors turnaround too. Yeah.
What other organs can you grow Androids from?
Well, I think so the mammary gland, like there's, I believe Boning is doing something very similar. Now, if you can classify that as an organoid, but yes, a lot of people are taking tissue from the udder of a cow and doing this work, but I think a lot of tissue you can do this. Yeah. What we're intrigued by is what we want to know more about, and what Koki will be trying to figure out is the day of harvest, the age of harvest, the breed. How does this affect the actual Android structure and function? In addition to testing some of these in vitro models like heat stress LPS and absorption of different nutrients and bioactives. Yeah. Tony?
Regarding the work with antimicrobials and probiotics, what do you think are like future directions for their industry practices based on this research, or maybe compared to what's currently like previously recommended? Or
Yeah, I think the biggest one from the bus work is to critically evaluate how much antimicrobial is currently being used. So I think we didn't really show a lot of these results, but when you're feeding this prophylactically, although it's not that expensive, you're really increasing your active dose of antimicrobials significantly through the entire production process of that animal's life, even if you're using it for 7 to 14 days. So I think that work from a practical sense, where I see it being applied is people are being more prudent or second guessing kind of, yeah, I know it's so inexpensive, but there is this, consumer perception we need to be aware and actually calculate in our return on investments in some of our research. So I see less use. With respect to probiotics, I see it being strategically used more. Instead of using it as a blanket across many different animals in the barn, maybe precision feeding it or offering it only to set amounts. And now that we're going to get really good at in the next 10, 20 years feeding animals individually, I see that that's probably the direction we're going to move into. But I still think there's so much more that we need to do in just characterizing the host more. And actually, of all the work that we've been doing in our lab, when we characterize the microbial community, we're not finding as much as characterizing this host. So I know I sound like a broken record right now with respect to that, but I want to make sure it's very clear, our direction. Any other question? So I'm going to teach my last first year class for this semester. Latero is leaving pretty soon. We have a whole bunch of the fruits of microbial fermentation here so that everyone can just respect and appreciate microbes and you can interact with them as well. And they're here, and Latero and Florencia will be here to answer questions. Koki needs to go to sleep for sure. But yeah, feel free to appreciate microbes.
Thank you very much, Mike. And your lab. So that's it. Come have a drink with Mike, and we'll send out the next invite for the CNM seminar in March. So thanks a lot, guys, and thanks to everybody online as well.
Yeah, awesome. That's good. See, it's funny how it comes together, eh? Thanks, Koki.
Yeah.